Well, thank you, Julie, for that lovely introduction, and thanks everyone for coming tonight. It's nice to see a lot of familiar faces. It's nice to see some new faces. What I would like to do, but seemingly cannot, there. What I'd like to do tonight is give you a brief tour through a topic that is actually really important for IHO. It's sort of baked into the DNA of IHO, and that is the importance of not just field but laboratory research. Or the way I sort of see this and the way many of my colleagues here see this is this sort of synergistic nature, this two-way street between going to the field and doing fun things and hopefully finding amazing things and coming back to the lab and trying to make sense of the things that you find. It's a bit slow. There. So for many of you, this may be your first time here at IHO, and so I thought I would just take a second to tell you a little bit about what we do. I think most of you know, I really don't need to go into great detail, that IHO was founded by Don Johanson and then sort of very capably captained for many years by the late Bill Kimball. And their work really focused in a lot of detail on sites here in East Africa, in particular Ethiopia, namely Hadar. You saw that in the video. But since the arrival here at ASU, IHO has really expanded into this sort of rogues gallery of really amazing colleagues and researchers who are really engaged in the study of human origins writ large. So all the things that make us human, make us unique, we have people here on staff who do it. And of course, none of us would be able to do anything without the amazing IHO staff that we have. So thank you. And collectively, IHO scientists now do work on almost every single continent on the planet. So we really do have quite a global reach in terms of our research foci, lab, field sites, et cetera. Also, some of you may be new or your first visit to our new building, the Walton Center for Planetary Health. And maybe you know, but maybe you don't. IHO occupies the entire second floor. So all upstairs, it's all IHO all the time. So there's people in labs working, and students and grad students and postdocs. It's kind of an energizing place to be. And what all of us are really interested in is this sort of transdisciplinary exploration of the origins and the ways in which humans are unique. And I want to talk about some of those things tonight. Again, like I said, most of you are familiar with the history of IHO and the field work that's been done, predominantly focused in East Africa and Ethiopia. And so here's a recent picture from one of our colleagues' field seasons out there. Of course, it has yielded over the many decades that IHO scientists have worked there many amazing fossil discoveries. These fossil discoveries are still ongoing. People are still in the field, even right now, as Julie mentioned. Johannes is in the field. I think a couple of other people are also over in East Africa. And this all got started, of course, with Don's amazing discovery back in the early 70s of this remarkably complete, and it turns out fully bipedal hominin nicknamed Lucy. It dated to just over 3 million years ago. If we try to put Lucy on a timeline, a family tree, that is, of course, much more in depth and much more fleshed out than it was back when Lucy was discovered. You can see here, this is where Lucy would be, a representative of her species, Australopithecus afarensis, that lived from about 4 million years ago and went extinct at around 3 million years ago. There are many, many different species. Who's more closely related to whom? These are things that paleoanthropologists fight about all the time. I don't really want to get into those discussions, but there's an amazing array, a tremendous radiation and diversity of early fossil human relatives. What I want to do tonight is just focus in on one part of this tree. Starting at just under 3 million years ago, I've colored in blue all the species that are contained within the genus Homo, the same genus to which our own species belongs. We are Homo sapiens. You can see us right here. And the species that are grouped in our genus Homo are fundamentally different from those that are not, from those that are in the genus Australopithecus or Ardipithecus. And I don't want to really go into a long laundry list of what those traits are. I want to focus in on one fairly long-lived species of early Homo, and that is Homo erectus, sometimes called Homo ergaster, depending on what part of the planet you're working in. We know from a fairly good fossil record that Homo erectus or Homo ergaster dates back to about 1.8 million years ago. We also know from the fossils of Homo erectus that it's one of the best represented species. We have complete skeletons, a remarkable understanding of its anatomy and behavior and cognitive abilities and lithic technology. We also know that it was the first hominin that decided to leave the continent of Africa. And so, as you'll see in a moment, we have representatives of this fossil species all over present-day Europe, all the way to mainland Asia and even present-day Southeast Asia. So what I want to do over the next 15 minutes or so is tell you a little bit about when and where do we see the best evidence, the earliest evidence for our genus's best represented species. And then I want to use that as a jumping off point to show you how some new analyses that take place in the lab can really shed some light on our understanding of what it means to be us, what it means to be in a species that's in this blue region here of this family tree. So I want to tell you a little bit about some field work that I've been doing since 2019. Of course, we've been interrupted for a few years by the COVID pandemic. This is, of course, a big multidisciplinary team. Not even everyone's listed up here. Principally driven by Stephanie Baker and Andy Harries. We've been working at a paleo cave site in South Africa, about an hour north of Johannesburg. That cave site's called Drimolen. And it's maybe a little bit difficult to see, but there's lots of sediments where various members of the team are digging, and various limestone walls, which were the ancient limits of this cave when it was either occupied or being filled in with sediment. South Africa has yielded many, many remains of fossil human ancestors, and the difficult thing of working in South Africa compared to East Africa is dating the sites. These are all old cave sites where sediments get really jumbled up. It's really hard to provide good dating estimates, but providing a good chronological framework is really critical to being able to say something about where a species may have existed on that family tree that I just showed you earlier. This site, Drimolen, is now one of the best and most securely dated cave sites in all of South Africa, at least in this time period. And I will just ask you to remember that age range, circa 2 million years ago. It turns out that that's quite important. So here are some excavations from one of the summers I was down there working. You can see several of us sort of keenly observing things coming out of this wall. We call it the Western Wall here. And this is me working on that sediment, that column of sediment here in the wall. And what was pretty amazing is that over the few seasons preceding and sort of culminating the season I was there, we found pieces of this beautiful brain case, this top of the skull. So this would be the front, the brow region. This would be all the way in the back of the skull here. And this skull cap came out of the ground in over 150 pieces. And it was really only the heroic work of my colleagues, Doctors Angeline Leese and Jesse Martin, who painstakingly over months and years put this skull cap together piece by piece. It was really a Herculean task. But it was worth it, because if you look at some of the details, and so here are various views of these skull caps, and I've sort of grayed out the detailed anatomy because you shouldn't be worried about that. That's for myself and my colleagues to argue over. We found all kinds of interesting components of its skull anatomy that told us it possessed all the telltale features, all the hallmark features of Homo erectus. And it was a real perfect fit for what a Homo erectus skull cap should look like. And once we sort of realized this, my colleagues and I, we decided, well, we have to run this by the world's expert in cranial anatomy of hominins, who was famous for sort of always making sure that one's anatomical ducks were in a row, so to speak, before he would give you his approval. And so we were very happy when this received the Bill Kimball seal of approval that this indeed was Homo erectus. It had all the telltale features of Homo erectus. So we got a thumbs up from Bill, which was always nice. Now, what was really important and interesting about this skull cap, aside from things about the anatomy, is that if you look at our site here in northern South Africa compared to the global distribution of other Homo erectus sites, something pretty interesting jumps out. As I mentioned, it's the first hominin where we begin to see representatives all the way out here in Western, now present-day Western Europe, all the way in mainland Asia and down into present-day Southeast Asia. And if you look at the best dates that we have for some of these sites, the site of Drimolen is older than any of those sites anywhere on the planet by potentially as much as a few 100,000 years. And so this Homo erectus specimen from Drimolen is the oldest representative, the most ancient Homo erectus specimen that we have in the paleoanthropological record. And so that makes it really interesting and really exciting. We don't just have Homo erectus from that site. I wish I could tell you more about the various different kinds of hominins that we have preserved at Drimolen. And we don't just have adult specimens. We actually have lots of specimens of juveniles, of young individuals, and infants even. And in fact, these are two bits, maybe it's a little hard to see, these are tiny bits of the front of the face. We call them baby faces because they're infant faces. And so my colleagues and I have been trying to study the anatomy of these infant faces and how the infant anatomy might have grown up to look like the adult anatomy, something about the growth of these fossils, which is one of my principal interests. And so, given that we have lots of young individuals and we have opportunities to study growth, especially in the fossil record, I wanted to just take a moment to highlight for you why the ability to study something about growth, something about development, or what we call ontogeny, is really critical. And so if we take a big step back and I was to ask you to sort of survey the room, what does it mean to be human, modern human? What makes us us fundamentally? I think, given the crowd, you'd probably come up with all the key features. And if I just run through them fairly quickly, you all are aware that we have exceptionally large brains. We run a really powerful computer in our heads. We are committed, or what's called obligate bipeds. We get around the planet in a very bizarre and interesting way, but it's freed up our hands to do a lot of interesting things. We manufacture and we use really complex tools. We're not the only ape that uses complex tools, but the nature of our toolkits are fundamentally different than anything else. We engage in all kinds of crazy creations of symbols and rituals, what you might call culture, and we even transmit that information, like I'm doing, to other individuals or in classrooms down through the generations. All of these are part of a complex culture, an intergenerational transfer of information that, again, humans possess unlike any other species on the planet today. We exhibit extremely high levels of cooperation. I'm not aware of any other species that's propelled another member of itself to another, the surface of another, to the moon or another planet. And to do that takes an extraordinary amount of cooperation. Tens of thousands of people working in concert. But also from a biological perspective, it turns out the way in which we grow up is extremely interesting and unique among mammals. We grow up very slowly. We take a long time to reach sexual maturity. We live, thankfully, long lives. And in that long life, we produce many, many offspring. We have a high rate of fertility. And this is what we in the world of growth biology call a unique life history. And modern humans possess this. And it's been argued in the paleoanthropological literature for decades when that weird modern human life history came about. And I've been fascinated with that question for a long time. And if we just zoom in on that one key area, this life history panel, one of the things about our life history that's really bizarre, that you wouldn't expect, is that we wean our offspring exceptionally early. For a primate, for an ape of our body size, our brain size, you would expect we would wean our offspring at 5, 6, 7 years of age, but we do it really, really early. That's what we call accelerated or early weaning. And it turns out that that accelerated or early weaning is one of the key features that allows all of these other life history components to occur. And so, as a paleoanthropologist, I've been interested in trying to ask, well, when do we first see early weaning? Can we see evidence of accelerated weaning? It's tough to do in the fossil record, despite the fact that if you go to the various museum collections, we have many, many fossil individuals preserved. Here's an individual from the same species as Lucy that died when they were infants, or really young kids. There's baby teeth just coming into the mouth. Here's another species from South Africa. You might recognize the famous Tong specimen. This year's the 50th anniversary of the discovery of Lucy. Next year's the 100th anniversary of the discovery of the Tong child. Died very young. There are other individuals that also died young. We've got lots of fossils. Now we also have lots of fossils from this new site of Drimolen, which preserve lots of infants and lots of young. So there should be some opportunity to say something about growth and development and maybe weaning. And it turns out there is. And the key is you have to know something about teeth. Now, all of you are familiar with the fact that if you went out on campus somewhere and cut down a tree, you would, and please don't do that, you would see something that looked like this. There would be these concentric rings that are embedded within trees. And it turns out plant biologists long ago worked out that for many, many trees, they deposit one ring per year. There's an annual rhythm to these growth lines. And so here, maybe you can't see, but this tree died after 24 years of growth. You just simply count up the annual rings. Well, it turns out that the components that make up teeth also grow in this incremental nature. And so this is just a microscopic zoom in. But the lines that are preserved in teeth are not annual. They're on a much, much faster schedule. And we can use that or leverage that information to say something about growth and development. So upstairs here at IHO, in one of the new labs that we have, we are running a very fancy micro CT scanner, similar technology to hospital CTs, but it's shrunken down and used for research purposes. We scan a lot of teeth. Here's a 3D reconstruction of a tooth that helps us figure out things about the internal anatomy so that we can section it and cut it in exactly the right plane. And then we can zoom in using all kinds of fancy microscopy. And this is a bit of the outer enamel, but this is a view of the inside of the enamel coating the tooth. And maybe you can see where these orange lines are. There are all these little tiny lines. Those are growth lines, but they're not annual, they're daily. Every day of your life is recorded in your teeth, in every tooth that's in your mouth right now, and in every tooth of every hominin and every mammal that's ever existed on this planet. So there is this amazing preserved internal chronometer. And in fact, these are images not from a modern human or a modern ape. This is from a Southeast Asian Homo erectus tooth, and this is from an East African weird, robust Australopithecine. These lines are all preserved. And that's really helpful, because if you look again, here's just a cut through a tooth. Here's the crown, so the part of the tooth that you chew with, this is cut in half, and the root hasn't fully grown. But because we can see every day in the life of a tooth as it's growing, it's very easy to use those daily lines, which also exist indented here, to say, okay, this area was at 87 days after birth, this area was almost a year after birth, this bit of the tooth finished at nearly 800 days, etc. And for this specimen, you could figure out exactly the number of days it was when it died. Just a remarkably high resolution image into the life of this individual that possessed that tooth. Now, how do we use that to say something about weaning? I teach a lot of courses here at ASU. This will be the shortest course I've ever taught. A real short course on environmental geochemistry. There are lots of what are called alkaline earth metals that exist in the soil all around us naturally. Things like strontium and barium, maybe you've heard of those. Barium is used for lots of medical purposes. And those break down from weathering bedrock, and they get absorbed by plant roots, and those plants use that absorbed water to create fruit, which animals eat, and then perhaps transmit that chemical signal to their offspring during breastfeeding. So every organism that's consuming plant life or some component of plant life is getting that chemical signal absorbed and passing it on to their nursing offspring. And it turns out that barium and strontium are really interesting. So they get incorporated into all the developing tissues of that growing offspring, their bones and their teeth. And we can use that information because we know, given some of the complicated chemistry, that things like strontium start out really low when animals are breastfeeding, but when they are weaned and they have an adult diet, strontium levels really increase. And the opposite's true for barium. And that's chemistry we don't really have to worry much about. But what it means is that maybe if we don't just glimpse these growth lines in teeth, but the chemistry that's linked to those growth lines, now maybe we could say something about weaning because we have a really good physiological model. So I've been working with some colleagues in New York, in Australia, and we are using a very fancy technique with a horrible acronym, laser ablation, inductively coupled plasma mass spectrometry. Rolls off the tongue. What we do is we take those cut teeth, we mount them in this chamber. It is passed under a laser that just rasters across the entire surface. And every time it rasters across, it sort of, with the laser, pulverizes a little bit of tooth. It creates a cloud of dust. And the mass spec machine pulls that dust up and instantly, at that nanosecond, records all that chemistry. And what gets spit out the other end, so here are a couple of portions of teeth, what gets spit out are these really high resolution biogeochemical roadmaps. And you can see there's lots of information, right? There's lots of banding, there's different colors, but there's clearly some banding, there's some component of time here. So we've learned now, this is just an example from another project on a population of chimpanzees. Here are some molars that we extracted from museum specimens, and here you can see these are just the strontium maps. We know the growth record from all those lines. Can we match that to the chemical record? And it turns out if you spend too many hours down the microscope like I try to do, you can see a change from very low levels here in blue and green to much higher levels, sort of glowing red hot. And we know exactly when that occurred because we could count all the days. And that dietary transition, which was weaning, occurred at just over or around 5.5 years of age. Even this is for a museum specimen. Nobody ever observed it in the wild. This is around when we think that population of chimps was fully weaned. So we have some good models. Can we say anything about weaning in the fossil record of our species? Well, we can. I can't tell you exactly what hominin we're working on, because this is work that's about to come out soon, so you have to be careful. But maybe it's a hominin from a site that I talked about 10 minutes ago. Maybe. We're going to edit that out, right? Yeah. Okay. We did the same analysis, so here are a couple teeth. So these are the actual fossil hominin teeth, and these are their barium and strontium roadmaps. And I think, I don't have to convince you very hard, that there are changes that go from high red to low and low values to high values. We can see that transition in the chemistry. And the age at which that transition occurred is coming soon. We're actually still kind of figuring it out, but I think we have a good estimate of that. Now, what we're doing is we're actually trying to work this out on many, many different species. So we have a grant currently in progress. We have members of earlier species of Homo, Homo habilis, our friend Homo erectus, these robust Australopithecines and gracile Australopiths from South Africa, Lucy species. And so we're actually busy collecting data to try to build up an entire portfolio of the evolutionary history of weaning. And what we'll be able to say is, if we think about the relationship of modern chimps to ourselves, we share a common ancestor anywhere from about 6.5 to 9 million years ago. What we're going to be able to hopefully understand is, when did that one key fundamental piece of what makes us, us, from a biological, from a growth perspective? Did it occur very early after that split, very late, so very recently, or at some point in the middle? And it's only because we're able to marry up this really fancy, amazing, lab-based approach with lots of colleagues, environmental chemists and field geologists and all kinds of people working at various disciplines, to be able to look at the fossils that we find in the field, not just with a new eye, but with multiple sets of new eyes, everyone contributing their expertise. And that's been the tradition here at IHO. We bring as many bits of evidence to bear as we can to try to illuminate the story of our origins. And so with that, I'd like to thank you for your attention, and I'm going to hand it over to Helen, or to Julie, sorry.